Hello, everyone. I would like to welcome you to today's Gateway to Translation webinar, where we have Dr. Matt Pearce from Medtechtomarket speak about from lab to clinic, how to launch a commercial diagnostic. And the Gateway to Translation webinar series hosts monthly events around topics of translating research into the clinic, and it's a joint collaborative by the University of Manchester and the University of Cambridge. Now, to start with, I've been asked to tell you a bit about me. My name is Dr. Claudia Lindner. I'm a senior research fellow in Henry Delford at the University of Manchester, and I'm also the translation lead for the Christopher Pankhurst Institute for Health Technology Research and Innovation. And my research is in the area of translation in medical imaging, where my group develops software tools to automatically extract meaningful information from medical images. And specifically, we are working on developing medical imaging solutions with regards to musculoskeletal disorders. Now, some of our work is on population-based studies, trying to better understand diseases such as osteoarthritis, but the majority of our work is focusing on developing solutions to support our clinical colleagues in practice. Now, in my role for the Christopher Pankhurst Institute, I'm focusing on creating a supportive environment to improve the translation of research findings into benefits for society. And here, in collaboration with Translation Manchester, we have developed and recently launched the Pankhurst the Pankhurst Health Technology Translation Toolkit. And this is an online resource that aims to guide you with regards to developing healthcare technologies. And it got a particular focus on a translational roadmap for medical devices that provides advice on the steps and activities to take place from the very early stages, from the initial research to something that is being used in the real world. And the tool comes with a whole range of additional features, such as a progress tracker and case studies. Now, without further ado, it's a great pleasure to introduce Dr. Matt Pearce, who has over 3 decades of experience across medical diagnostics and biotechnology. And Matt is the founder of Medtechtomarket, which is a contract design, development, and manufacturing organization for diagnostics and medical devices that spans the entire product life cycle. So all the way from very early concept stage to a product that is ready to be used on the market. Matt, over to you.
Hi, good afternoon. Thank you, Claudia. So, as you mentioned, yes, more than 3 decades. About me, 3 decades in the IVD industry, across a wide range of roles. And over my career, I've probably been responsible for more than 50 million diagnostic tests worldwide, and 30-odd thousand instruments, with a range of diagnostic companies. I set Medtechtomarket up about 13 or 14 years ago, and we at Medtechtomarket, we're basically an outsourcing solution and translational partner for medical and diagnostic technologies for a global market. We're based, just quickly about Medtechtomarket before I get into the detail. We're based up in the northwest, near Liverpool, and we also have offices in Ireland that will allow us to do the regulatory submissions for European Union and do our own EC representation. Within the UK, within our head office up in the Wirral, we have all of our development labs, biohazardous production labs, as well as development labs, warehousing, etc. Okay, so on to understanding how to develop from lab to clinic a diagnostic system. I'm just going to give you a quick overview of the regulatory framework. I've only got 40 minutes. I haven't got time to go through all of the processes and the detail. Certainly happy to follow up afterwards. And then going through the development processes and activities, and then all the way through manufacturing, clinical performance, labeling, submissions, and then some at the end. I'm going to just wrap up with a few commercial considerations, which you should be, although I put these at the end, actually these should be at the beginning, looking at the planning for the territories, what the commercial models are for diagnostics, and how you go about costing, etc. I have to warn you, some of the slides have got quite a lot of content. I've done that deliberately because they're going to be made available. These slides are going to be made available through the translational toolkit, so that you can pick up the information later. Okay, you've been warned. Right, so the first question is and this, surprisingly, this happens a lot. We're presented with the question is this a medical device or not? And then from that, you can then decide whether it's a diagnostic. So I've pulled out here, and I'm going to basically wrap the regulatory standards up as quickly as possible, but to give you the essential information that you need. But basically, within Europe, the medical device regulation and the in vitro diagnostic regulation are in place. They're very closely related. Obviously, IVD is focused on IVD. So I've put here excerpts from both of those standards to allow you to, if you're looking at a technology, to understand whether it is classed as a medical device, because there is obviously borderline and technologies, wearable devices, or whatever, that may or may not be considered diagnostic or medical device. But basically, the key bit here is the words that I've highlighted in bold. So if the product that you're developing or technology is an instrument, appliance, software, reagent, or material, or anything that's used either on its own or in combination for human beings, that's a really important statement. And if that's being used for the purpose of diagnosis, prevention, all that, those in bold, then it is a medical device. And the blue at the bottom is specifically for the in vitro diagnostic, is basically saying if this is being used in vitro for the in vitro examination of tissues derived from humans, then it's an IVD. The US has very similar definitions within their 21 Code of Federal Regulations, and UK MDR, which is the UK standard, that also has very similar. The medical device software regulation, MDCG, is 2019. There's also a 2021. They again pick up on the MDR side of things. So software that also is used for this specifically is part of that regulatory framework. So diagnostic tests and diagnostic systems doesn't necessarily mean in vitro. It could mean a medical device, and so there's a spectrum. So, and this presentation is sort of going to cover everything really, whether it's on the medical device end of the spectrum, and that's like the glucose patches or heart rate monitors, pulse oximeters or whatever, all the way through to the other end of the spectrum, which is more traditional in vitro diagnostics, whether that's laboratory equipment or lateral flow. It's all IVD. And that also means packs of reagent kits for running on ELISA plates or dipsticks or whatever. And then somewhere in the middle, there is also, and these are accessories to the IVDs, things like blood collection kits, PAX gene tubes, that sort of thing, but also interpretive software. So these are all of these things here are medical devices. It can be quite nuanced in terms of what the intended use is, and I'll come to that in a minute. But pretty much, if it falls in those definitions I mentioned before, then you're in this spectrum. Okay, so typically within a diagnostic system, there are chemical elements, and that's whether it's PCR, immunochemistry, lateral flow, whatever. Chemistry aspects, unless it's a solid state type test like an ECG. There's software, and so it could be all or some of these things. There's software which can either be embedded or application based. Electronics, there's calibrators. So these are often human origin calibrators, so freeze-dried plasma, for example. There are also other types of calibrator, sample collection devices, I mentioned, and then various connectivity options, and also other accessories. So let's get into the development process. So the product development process, I like to use the analogy, it's like making a cake. The actual process and the sequence of making that cake is pretty constant, but also the ingredients are project specific. Now the process, the sequence is fairly constant, but also critical. So if you bake before you add the flour, then obviously you don't get a cake. So the sequence is really important, but also the ingredients are what actually makes up the functionality of your test and your system, and you've got to get those pieces right. And so really the way I'm going to focus the next few slides is really about during the development process is how do you focus on what is the process and the sequence of events, and then specifically looking at the ingredients and what needs to go into the diagnostic product. So in terms of milestones, many of you will be familiar with the technology readiness level scoring system, which basically takes you through from 1 till 10, which is sort of launch. And that and I've aligned that with what I just class as typical milestones. And I've broken these down, and because the TRLs isn't always a definitive numbering system, I've just broken these down into stages. Now, the fundamental difference between a diagnostic system, product development and academic research, and bearing in mind the purpose of this talk is to where do you go from taking an academic identified biomarker or technology and turning that into a product. In academic research, the real focus is on the two pieces that are highlighted in red. So the research program, obviously, the technology discovery mode, biomarker discovery, and then clinically validating that in the clinical validation program. Now, in terms of product development, the fundamental difference is that you have to demonstrate that the product is safe and effective, not the technology or the science. And so, again, just like baking a cake, the sequence of this is very critical. So there's a whole load of stages in between which you need to go in this order in order to sequence to deliver this program. So it is you cannot jump straight to clinical validation on the basic technology and use that data in a regulatory submission. You have to go through the proof that this is the product that you are validating with that clinical studies. I'll talk about clinical studies later. Just a really quick overview. Again, I'm trying to just do this because you'll have heard these expressions, so just a basic oversight. So competent authorities, which are things like the FDA or MHRA in this country, or within Europe, they have their own agencies like the HPRA in Ireland. They dictate the regional requirements and the registrations that allow you to commercialize have commercial activity within the territory. So in the USA and the CE marking, that means you do need to you require a technical file for most in vitro diagnostics. So in Europe, it's roughly about 80% of all diagnostic tests need to be technical file reviewed, and in the US, there's varying degrees that you'll hear these expressions 510K pre-market authorization or de novo. That just depends on what your technology is and how new it is and how novel it is. You also need to register your product as well as the establishment within these regions. In order for Europe to actually do that technical file review, in Europe, the MHRA don't necessarily do that. They use notified bodies, and these are companies that specialize in reviewing the technical files. So this could be BSI, DECRA, TUV, UL, whatever. And there's about, I think, 10 or 12 of them throughout Europe that will allow you will do technical file reviews for diagnostics. In the UK currently, as of January this year, the UK for most diagnostics, not all, are self-declaring and under the general classification rule. And so that's a slightly different, so that's just a registration process. Okay, so in terms of the essentials, so in order, and I'm still focusing on the process. So before we get into the actual the process of those individual milestones, I still have to just give you one last oversight of the regulatory framework in which we operate. But basically, all medical device manufacturers, and the word manufacturer really means owners of the product design or the economic activity, they need to be certified with a recognized quality management system in order to submit for IVDR review. That really means ISO 13485, which is the quality management system that governs the development, manufacture, and commercialization of medical devices or in vitro diagnostics. And that is really a framework of procedures that you have to be compliant with so that everything has been done properly. So you've trained your staff, you've calibrated all your fridges and your pipettes, etc. You're following a procedure for design and development. So that's just an overarching quality management system, a bit like ISO 9001, which is a generic quality management system. But within medical devices, there are also 4 other standards which are also mandated in order to launch your product. And that is the risk management standard, a usability standard, so you have to consider how your products are going to be used, and then 2 relating to the instructions and the symbols and the labeling that you use on your product. Everything else, and there are hundreds of standards, all other standards that govern the safety and efficacy of the product, they vary depend on the product, of where it is used, how it's used, who on and who by. And collectively, we just call that the labeling claims. So this is where you have to provide evidence against the standards. And now I'm going to talk you through how you identify where those come from. So the whole project of all diagnostic projects, all medical device projects should start with an intended use statement. This is a really important document, and you can see this on open up a pack of paracetamol or open up any diagnostic test, and you will have in the instructions for use, probably on the first or second page, there will be an intended use statement. And here I'm just using 2 examples. On the left is a glucometer. They're both glucometers, so glucose readers. One's from Roche, one's from Johnson & Johnson. And these are just typical glucometers. And I've exerted the intended use statements. And I've highlighted on the left what the intended use is. And as I mentioned to you, the intended use you have to consider about your technology is how is it going to be used? Who is going to use it? Who are they going to use it on? What are they going to be testing? Where are they going to be testing this? And are there any kind of exemptions or limitations? So I've just chosen glucose because it's a relatively straightforward thing for everybody to understand. So you can see there I've highlighted within that intended use statement how that one statement is addressing all of those areas that are important. And it's important to stress here, if you look in red, it's the fresh whole capillary blood samples drawn from a fingertip. It's being used by people in orange by a single patient, so it's intended to use by a patient for self-testing, possibly with their healthcare professionals or caregivers, that really means, in the clinical setting. But then in orange, as you see at the bottom, it's only intended to be used by a single patient and should not be shared. And then the purple shows you what it's being used for. So this one should not be used, for diagnosis or screening of diabetes. On the AccuCheck side, on the right-hand side, I haven't bothered highlighting it, but you can see here there's a whole range of different things. This one's actually for quantitatively measuring glucose in venous blood, arterial blood, heel stick neonatal, fresh capillary blood samples from the fingertips. And then it also says who it's to be used for, multiple patient use. So fundamentally, the evidence which you need to deliver and you need to demonstrate in your technical file for these 2 products, whilst they're both glucose readers and they probably have the same level of accuracy or same level of glucose performance and probably very similar chemistry, the evidence you need to demonstrate these 2 intended use statements is very different. And this is where the ingredients and the subtle nuances are. So if you take anything out of this presentation today, the most important thing you can do when you're considering your technology is to create an intended use statement, and because that's where it all begins. So in order to write an intended use statement, you need to really understand your clinical setting and your care pathway, and engage with some clinicians to ask them how a technology this might work. Understand the market and the market research. So where is to inform your launch plan, because sometimes your intended use scope will be tailored to how you intend to launch the product. So maybe you wouldn't launch this for home use to start with. You might launch it for a lab use. It's slightly easier to prove. So things like that, but also review any competitor or similar products. Look at their instructions, see what they've written, because that gives you some guidance. And then the final piece of advice I always have is don't get too excited at this stage, because you need to keep your intended use statement manageable and realistic. This is a traceable thing. You should write this at the beginning of your project, and all of your design and specifications, everything will feed off that, as you'll see in a minute. So keep it realistic, as you'll need to provide evidence against that to show that you are meeting your intended use. So once you have the intended use statement, you then use that to guide your design inputs. As I just mentioned to you before, some of those things you would have to validate fresh finger stick blood, or you'd have to validate heel prick blood, or venous or arterial blood. So all of that will be built into your design specifications, which we call design inputs. So this is where you identify all of the different aspects of the product based on that intended use. So robustness, where is it going to be used? Who is it going to be used by? Robustness to shipping. If this is going to be used at home, you have to consider how you're going to ship it to people's homes. And that also features in your validation and your design specifications. And so that covers all aspects of the safety, the power use, labeling, where it's used in normal conditions. Is this stability? All of those aspects need to be covered in here. And you will create a design. You'll need to create a design inputs document, and it's just a list of specifications that you list, that you assign with an acceptance criteria of what would be acceptable. Design intent and traceability is extremely important. FDA particularly have very strong link on this. They will ask you in a 510K submission to send them your design inputs, and the traceability is your design outputs. So you demonstrate where you've designed all of these aspects and you've met your labeling claims. As well as the design inputs, you have to consider risk. I mentioned about risk management, ISO 14971. Basically, risk assessment is understanding, once you know who your customer is, where it's going to be used, etc., you can then generate a risk management plan based on how severe the technology is, whether if it was a misdiagnosis, how much hazard or how much harm would be caused, what the likelihood of that is. And you list all of those risks that you can foresee of it being misused, misdiagnosed, all that sort of stuff. And that document is a live document, but it actually might generate more design specifications, because you need to demonstrate during the course of a project, and this is a cyclic thing, that you actually need to revisit and show that you're designing out the risks, and then you have whatever residual risks. And again, with the FDA, you will be submitting a risk management plan and your risk assessment, and you will be listing out any residual risks that are left within the project. Okay, so once you've got all those pieces in place, you then need to start focusing on the design. So the first thing you should write in diagnostics is a performance evaluation plan. This is where you basically combine all of the design inputs, risk assessment, regulatory standards into a plan, plus how you envisage testing, and what your design process should be. I would also at this stage recommend you start planning your clinical trials, because the planning of those takes time. But whilst that's happening, you should also be getting into the chemistry and test optimization and the electronics design to maybe breadboard stage at this stage. Software development, so that's a lot of diagnostics or medical device systems are software only applications. So at this point, you'll be detailing out the architecture, the functional design, life cycling plans for the relevant regulatory standards for software, and also any algorithm development and training. So a lot of this work will have already been picked up from your research project and your discovery project, but now it needs to start to be consolidated you're now thinking of developing this into a product and firming everything up. At this stage, you'll also be looking at packaging and starting to thinking about how to productize the product. So at this point, depending on what the diagnostic system is, I'm covering this as if it's a full instrument and test cartridge system, but irrespective, even if it's an ELISA technology or a PCR technology, then you need to consolidate the chemistry, the plastics, the full mechanical design of everything that goes into that product, the packaging, any calibrators, you need to start designing those. And all of this should be documented, of course, with again, going back to design intent, you need to document what you're doing, keep a record of everything. And then all the way through to once you've got the hardware and software complete, you've written the software, you've done a final system integration. At that point, you can consider the design frozen. We normally at this point would also do some indicative performance confirmation. So this is where you test it against some of those design inputs. So even though it's a prototype at this stage, the design is frozen, but to satisfy yourself that the design is frozen, you test it against some of those specifications. So does it give you the right linearity? Does it give you the right precision? Does it give you some the right kind of test results on the algorithm, as you'd expect based on the work you've done, or the patient samples, or the data you're processing. Then at that point, for product-based technologies, you then need to go into manufacturing validation. It's really important that, as I mentioned to you right at the beginning, sequence is important, that the product is what you are validating. So at this stage, you need to demonstrate it's a product, you demonstrate that you can it's manufactured. And this is basically, you're representing what the customer will see. So the clinical performance and final verification needs to be done on the product. So from now on in the project, the manufactured product is used. If there's any design changes, and there often are design changes needed, you will then have to take a step back and revalidate and re-qualify those design changes. And that also includes checking back over risk assessment and checking back over your design inputs to make sure you haven't jeopardized your risk or your design process if you do make a subsequent design change. But at the manufacturing stage, you're really looking at setting up into place the raw. I mean, this can be outsourced, of course. You can set up your raw materials, your suppliers, assembly jigs and fixtures, kitting processes, all your QC testing processes and specifications. And then also you will do what they call a process risk assessment. So that's an FMEA, which is basically looking at your manufacturing. Where could it go wrong? And making sure you validate that away. Typically, you would at this stage produce multiple lots, normally 3 lots of product, using variable raw materials, variable operators, etc., and qualify those as performing as they should be. But you can also use those 3 lots for your validation, your verification and validation. So at this point now, you have a product that looks and feels like the real thing. It's in its boxes. It's been dispensed by the manufacturing team. And to all intents and purposes, this is the same as what the customer will see. Maybe not with the branding, but it will have pretty much everything else in the pack or in the kit. So this point now, you start to look at verification and validation. And this is where the detail of this, and I have to be fairly vague because it all depends on the product. But if you go back to those intended use statements, you can imagine the kind of validation that you need to do. So at this point, you would write validation protocols. So you list out all the studies you're going to do as a protocol. Again, design intent, very important. What the acceptance criteria is, which is picking up from your design inputs, and then performing the validation itself. And things like stability studies. So if your design inputs say it needs to have a shelf life of 12 months at room temperature, you make sure you have that data to support that. And normally you support it for 13 or 14 months, and then you have 2 consecutive time points that show stability, and then that way you can use the first one of those as your cutoff. Stability life studies, so that might be study an instrument, and in fact one of those big differences between those 2 glucometers is that the decontamination and the life of that expectancy of those instruments, because one of those isn't so robust to repeated wiping, which is why it's single operator only, because you don't have to worry about cross-contamination of blood. So you have to do life studies to demonstrate your instrument or your electronics are up to the repeated use. Also, then you have assay performance. This is really important. Linearity, precision studies. I'll come to some of these in a minute. And things like limits of detection, also obviously, and whether this is PCR or immunochemistry or whatever, or enzyme assays, you still need to demonstrate the assay performance. Interference studies, there are regulatory standards for interference studies, but also you need to consider your intended user, your intended use patient. They may typically have comorbidities that require you to look at additional interference. So again, you have to consider that. The operating conditions and workflow, open vial stability, for example, or if the test takes so long, is it valid? How long does the blood sample stay robust for? All that kind of stuff, to understand that the in-use conditions, you've covered as many of the variables as you possibly can. Distribution, I mentioned that at the beginning about who you're shipping to. Electrical safety testing. If your product is sterile, it's not particularly common, but it does occasionally happen. If your product is sterile, then you need to do sterilization validation. And there's regulatory standards specifically for that. You can look them up on the BSI website, for example. Usability, software validation at this point. And then finally, you'd also be at this point write a scientific validity report, and that's just showing all the evidence around your technology and why it's valid for clinical use. Okay, so once you've done the V&V, in parallel, you can also be doing your clinical trials. So these clinical trials are different to the kind of questions you would ask in an academic clinical performance trial. For this, the clinical trials is all about getting the data to satisfy that the product is safe and effective for use. You're not proving the biomarkers as such. This is about what you're going to submit. So the clinical trials vary, and I have to caveat this, they vary depending on the product type and the application and the intended use setting, for example, obviously. You need to run your clinical trials in an intended use setting. So if you're saying this is for use at home, then you need to demonstrate home use works. Now, there are different types of studies. If your technology is in a clinical chemistry application, then typically we do a method comparison study where you would test your measurement of known biomarkers against a predicate device, so against a competitor device, or a device that is already approved on the market, or and that can be a lab reference method. It doesn't have to be a near patient test. If you're developing a near patient test, it just has to be an approved lab reference method. Typically, you run 120 patients, so these aren't massive studies unless there's gender variability, of course. And in the UK and Europe, one site is okay for this. In the US, the FDA typically expect 3 sites, all of which ideally should be in the US. For oncology type applications, then it's more to do with sensitivity, specificity, and positive predictive value, negative predictive value, for example. And those you're comparing to your gold standard. You can do predicate devices as well, but also the studies and the size of the studies are powered by the prevalence and the confidence intervals, etc. So in those studies typically sort of 400 to 500 patients, but it can massively vary depending on the prevalence. In addition, there's other studies which may or may not be needed. There's a normal healthy reference intervals. So again, you need to demonstrate that your product comes out negative, for example, or below a certain clinical decision threshold for normal or healthy individuals. So you need to demonstrate that. So that's typically another 120 patients. These are healthy donors that could be used. And then there's a series of studies which are covered by their own regulatory standards for doing precision studies. And there's 2 typical ones. The mainstay one is what they call a 20-day study. And this can be done by the manufacturer at their site. And this is a study where you do 20 consecutive days of testing the same samples, both morning and afternoon, multiple instruments, multiple batch of product at different levels, depending on the cutoffs and the relevance. And so it's a big deal, these studies. And they can go horribly wrong if you don't focus on them. But there is quite good guidance documentation on how those are constructed. The alternative with the FDA, they allow 5-day studies, usually over 3 sites. And those are where they're running by an intended use setting by intended users. And they just basically do a truncated version of the 20-day study over a 5-day period. And in addition, you sometimes have whole blood or whole matrix precision studies where you have to demonstrate the performance is reproducible in blood. But that all depends on the application and what is feasible to get reliable blood. So just a couple of points, just a couple of loose ends to do with the clinical performance. For the de novo products with the FDA, so early in the project, it is highly recommended that you engage with the FDA through a Q-Sub process. The FDA and the MHRA, for example, do have an online support, and they will give you advice to allow you to structure your plan so you can approach them. But basically, if you are in that de novo product range, then everything needs to be agreed in advance with the FDA. So once you know exactly what your project is, you should be mapping everything out with them, engaging with the FDA. There are some other things. Obviously, software validation projects can be used. You can use validation cohorts of data. So this is patient data that's not being used to generate your algorithms, but is just being used to validate, provided that data has been generated in its intended use correctly from pathology samples or whatever, then that can be used. And then depending on your intended use, there are some other studies that need to be considered. So if you're a home user product, then you need to compare professional user that are trained or naive versus self-testing versus a lab reference method. So you're demonstrating your product actually is accurate to lab quality, whether it's a professional user or a self-tester. So that's the kind of study that you need to generate there. You can use virgin biobank samples again, but these need to be compatible with the intended use. So typically DNA and RNA, but these are rarely allowed as the only thing. Certainly with the FDA, they like to see that you've actually run intended use studies and proper clinical performance studies. Some studies, so you can also collect samples from the appropriate intended use setting and run those back in your lab. So that is also allowable in some cases. And so again, it just needs to be reviewed on a case-by-case basis. Finally, you then focus on during this. Now, I do advise to start this early. This always seems to take longer than you think. But basically, you also start to pull together your instructions and your labeling. So your instructions for use will contain all of those labeling claims you talked about on the performance, what the intended use statements are, and everything else. But you also use these instruction use for your usability studies. So you have to demonstrate effectively that your product can have an out-of-box training. So if somebody reads this operator manual, can they run your test? So, and if it's for professional use, that's fine. It's professional users who you need to demonstrate it for. So, so you need to consider that. Just as a point here that the intended use statement does also get added to the front of every box and also typically on the front of all of your instruction manuals. Languages for Europe, as I show you here, depend on what your commercial plans are, you need to consider your languages too. So for Europe, these are the languages, depending on what country you're planning to trade, you need to provide your instructions in those relevant languages. Typically for medical translations, you do a two-stage translation. You have the words doing primary translation, and then you get independently verified with a secondary translator. And usually medical translation companies will do this for you. Hard copies aren't essential, but you do need to provide easily retrievable e-copies. And unless there's a risk, and unless you've identified your risk that there is a problem if you don't provide the instructions. All of your documentation, your labeling will be submitted both to the regulators, but also to the EC representatives. So they will also check your labeling as part of your submission. And at the end of this, once you've finished this stage, to do all of that V&V and the clinical work, at this point, you've probably run between 40 and 50,000 tests. And remembering those processes that are underpinned by ISO 13485, you need to keep traceability. So every test result, every operator, the calibration, the samples that you've used, etc., keep all of that as records. Electronic is fine, or paper. We typically do electronic and paper, with the paper copies all double signed and checked. But everything needs to be traceable, because in the event of an audit, FDA, for example, will inspect and they will drill down to individual test results. And on that day, who was running it? Were they trained? Did they have calibration? Were the pipettes calibrated, etc.? So all of that traceability needs to be retained. But at the end of the day, you have a technical file which contains a number of different elements. Obviously, the device, the design inputs, as I've talked about, the specification document, but also all of your labeling, all your manufacturing and design information, safety, risk assessments, all your V&V and clinical performance data. And then the last few are the sort of traceability to that, show you where your design inputs and design outputs, and all your CE marking, just your alignment against the standards. So that's what they call an essential principles checklist, where you show all of your standards that you say you need to be compliant with based on all of the different aspects of your product, that you've met those relevant standards. And then finally, you draft up a declaration of conformity. And as an example, I use this photo. This is a founder who approached us a number of years ago. And in that photo, you can see a small document on the desk in front of you. That's the technical file that he gave us when he started the project with us. And the files at the back, and this was a class 1 medical device. The data was what the technical file is in those files behind. So that's really what at the end of the study, that's what this looked like. Okay, so into commercial. So territory considerations. I've got a few more minutes left, so I'll just quickly whiz through this. The UK is self-declaring for general IVDs. So that's 80% of IVDs are still self-declaring in the UK, which means it's relatively straightforward. Obviously, it is still assumed you have all of the compliance and a technical file in place. So you know, it's not just, don't just make it up. You do need to have this stuff, and it could be audited. But a word of warning that the UK NHS is very slow and difficult to get adoption and roll out. So for startup businesses, that's commercially quite a risky option. However, the NHS and the UK is very good for evidence building and research studies. Also, the other thing, specifically for diagnostics, the market is very dominated by the hospital lab market. There's very limited point of care testing done in the UK at the moment. And so if you're developing a point of care diagnostic, the UK isn't necessarily the best place to launch that product. CE marking, again, you have to, as I said, you have to review through notified bodies under the IVDR. But within, and that typically, in order to book in with UL or BSI, you also, I would recommend that you have your 13485 accreditation through the same notified body. So check with them first. But then also, there's about a 12-month lead time in order to book a slot for that technical file review. So book early, don't wait till you finish the project until you book that slot. Now, in Europe, launching, Germany, from my experience, Germany, Italy, and France are the earliest of the adopters. Germany particularly is good because it's very reimbursement driven. And there are really good EU distribution networks. You will need to have an EC representative who is legally responsible for your product while it is on the market. And for every territory, you will need to notify them that you are selling in that territory. In the US, the FDA approval process depends on the product and the level of risk, and whether there's anything else like it on the market already. So again, as I mentioned to you before, seek FDA Q-Sub advice. But do that once you have a clear intended use and performance evaluation plans. That's at that point to discuss with them what you're hoping to do, and also to get feedback on the regulatory. You can also seek a regulatory advisor, obviously, and get a regulatory roadmap produced. The US is 50% of the world's healthcare budget. It's very highly matured IVD infrastructure. There's a very good, strong network of distribution. There's walk-in diagnostics clinics, physicians' office labs are very switched on to using and early adopting, and there's a clear reimbursement, and I'll talk about that in a minute. Again, with this one, you don't need a lead time in order to submit your technical file for 510K, for example, but you should allow for around 12 months to gain that accreditation if you have all of those pieces in place. And I would definitely recommend you use an FDA advisor to help you navigate through constructing the 510K or the PMA documentation. Right, lab developed markets. I haven't really talked about that. There is also a lab developed test market, both UK, Europe, and the US. It's much more advanced in the US. There are companies who specialize in performing tests, and at the moment they are still outside the FDA. So effectively it's a way of gaining commercial traction in the US. You have to work through those what they call clear labs. So Quest or LabCorp are the two of the main market leaders. And so those guys may adopt your technology, and they will then help to effectively sell that as a service for testing those samples. In Europe, the LDT market still falls under IVDR, so you still have to go through that process of accreditation. So just a real quick thing. I'm not going to go through these in detail, so I don't have a huge amount of time, but how to launch. Obviously, you can license out technology. Just putting that to one side, you can launch I would always recommend you launch via distribution. And there are really good global distributors, but also very big if you're focusing in the US, for example, as I mentioned, Cardinal, McKesson, Shines, all those sort of people. And typically you're giving them between a 30 and 40% cut. They also operate a thing called reagent rental, which I'll talk about in a minute. And these guys are good because you can leverage their existing, their approved suppliers already to the health authorities or to the health and to the hospital trust, for example. They have their own sales teams. They've already got the reagent rental framework, and they also will handle your basic complaints. Then there's another area which is historically has been quite good, and that is to work with existing global manufacturers, partnering with them as an OEM distributor. In that case, that means you rebadge your technology under their name, or they do a global co-selling. And for that, they typically take between 30 and 50%, depending on the distribution and the type of product. That usually includes marketing, so they will do a lot of your marketing for you because it's effectively their product. Reagent rental obviously carries on, but also this gives you a lot more access to some of the benefits of leveraging their brand names, but also their complaint handling and regulatory affairs work will help to act. So, and also with these kind of things, it's usually a first stage to exit. Direct sales is a possible, particularly in the US. They have a thing called manufacturers reps, and these guys are guns for hire who might have 2 or 3 products that they're selling, medical products, or 10 medical products they're selling. That allows you to retain more profit because you don't give them such a big cut and you're commission based and all the rest of it, but it's risky, slow, and quite problematic. I would always recommend you consider distribution. Reagent rental is in the US. So if you have a product that's an instrument and a test cartridge, it is illegal under inducement law to give away the instrument because this is a razor blade model or the inkjet printer model. You can't give the printer away just to fuel your cartridge sales. You have to, and you can't do it below cost. So the way around this is they have a reagent rental where they basically reduce the cost of the capital spend by renting you the equipment and then you bill them for recurring costs of using the consumables. And you can often do that with fixed minimum order quantities, incentivized pull rate plans, or whatever. So that's how a lot of the diagnostic industry works in terms of commercializing. And that does allow you predictable revenue, which you can then use to get investment money or growth capital if you're going that way. You have to remember you need to buy those instruments because you're going to be placing them in the market effectively for free on the long-term rental. You need to consider your cash flow. And then finally, I've just got a slide here, just to bear in mind about costing. And this comes back to right at the beginning, I mentioned about doing some market research. You also need to do some health economics, and this should also be done at the front. But basically, you need to consider this model I've drawn up here is the US model, where the doctor who's prescribing the use of your diagnostic or the collection of blood for that test will take a cut. So if your reimbursement is $40 for that test, and you can look up reimbursement codes with cms.gov, there's what they call CPT codes, they will take a percentage of that. So they will then pay 24, let's say pounds, dollars, whatever, 24 pounds for that test. The distributor, and there may be one or two distributors in the chain, they will take their own cuts. And then eventually it will understand to you, and you will basically be selling your product for $11.76 for that product. So you need to bear this in mind, because your cost of goods then needs to be within this. And so ideally you're looking for, you want to try and aim for a 55. So you need to 50%. So you're looking for about £5.88 or $5.88 per test kind of cost. So that gives you an idea of, but you need to work that out. Now for UK and for some European countries, having a detailed health economics and a business case is also, you also need that to show that there's a cost benefit of using the test. So it's a slightly different way of costing your products. Okay. So on a typical development, so working out the return on investment, typically your project should take, or this is how long it takes us, but typically lateral flow test should take under 2 years for most relatively simple products, maybe 3 to 4 years for a more complex instrument. And then medical devices are usually a bit quicker, as are software applications. Funding routes. So some of the funding routes in the early stage, you can use translational grants to fund some of this work. In fact, a lot of the translational grants will pretty much take you up to, you just need to, the wording is careful, but will take you pretty much up to the point where your design is frozen and you are also prototyping it for clinical use. So that means you need the manufacturing, and the cost of the manufacturing validation is built into the actually production of those tests. So it does cover pretty much up to submission readiness. And academic grants will also fund some of this. So BBSRC will fund a lot of this kind of work as well. So it doesn't have to be a translational funding. So that will get a lot of this work underway. And then obviously, as you get further along the line, then you're starting to look at more equity investment, spin out or licensing out at that point. But there's different mechanisms of funding it from then on. So I think I'll leave it there. Thank you. And if there's any questions.
Thank you very much, Matt. That was really, really interesting. And I think a lot of useful advice there. And yeah, it's really great that you made the slides available alongside the recording, and we will put it on the toolkit. So I think it's really, really useful resource going forward as well.